Hello. So today we'll be talking about a question which came in GATE 2023 Biotechnology paper, and we'll be discussing the question. So the question is, in ELISA, which of the following enzymes are conjugated to antibodies for detection of the analyte? The options are alkaline phosphatase, trypsinase, horseradish peroxidase, and amylase. We'll talk about the technique of ELISA very briefly. So ELISA is the basic assay technique known as enzyme linked immunosorbent assay. It is also known as EIA or enzyme immunoassay, and it is basically carried out to detect and measure antibodies, hormones, peptides, proteins in the blood. Antibodies are blood proteins produced in response to a specific antigen. So it helps in detection of certain infectious diseases. So when there is heightened response, heightened presence of antibodies in the body, that means there's chances that that particular infection has occurred. So it is basically a distinguished analysis compared to other antibody assays, as it yields quantitative results and separation of non-specific and specific interactions that take place through serial binding to solid surfaces, which is normally a polystyrene multi-well plate. So why is it known as enzyme-linked immunosorbent assay? The antigen or the antibody of interest is absorbed on the plastic surface, that is why it's called sorbent. Antigen is recognized by specific antibody, which leads to the nomenclature immuno, and this antibody is recognized by second antibody which has enzyme attached, which is why it's called enzyme linked. Whenever we add substrate to it, that will react with the enzyme conjugated to the antibody, and that will produce a colored product that will give a visual readout. So a little bit about the history of ELISA. Peter Perlman and Eva Engel at Stockholm University and Anton Skurs and Pok van Wumen in Netherlands independently published papers around 1970s that synthesized this knowledge of ELISA into methods to perform ELISA. So basically, in the most common approach to using the ELISA technique, an aliquot of the sample or calibrator containing the antigen to be quantified is added to and allowed to bind with a solid phase antibody, and after washing, an enzyme labeled antibody is added and forms a sandwich complex of the solid phase antibody, antigen, antibody, enzyme. The unbound antibody is then washed away and the enzyme substrate is added, and the amount of product generated is proportional to the quantity of antigen in the sample. So specific antibodies in a sample can also be quantified using an ELISA procedure in which antigen instead of antibody is bound to the solid phase. So this is how a developed ELISA plate looks like. So this is the basic principle. So the principle is that specific antibodies bind the target antigen and detect the presence and quantify antigen's binding. So in order to increase sensitivity and precision of the assay, the plate must be coated with antibodies with high affinity, and ELISA can basically provide a useful measurement of antigen antibody concentration. So, and it's one of the easiest blood tests also that can be carried out. It's rapid, quick, and requires a blood sample of the patient, and the entire procedure can be followed out, done very easily. So in one case, what happens is that you can take different bodily fluids from different samples and basically add to the wells, and if you are trying to detect a specific antigen, the specific antigen will be in that well. Now you will add a primary antibody corresponding to that antigen. That primary antibody will go and bind to the particular antigen. Then you will add a secondary antibody. The secondary antibody will be specific to the primary antibody. The secondary antibody is not specific to the antigen, it's always specific to the primary antibody. For example, when we do it with specific proteins, for example tubulin, if you want to measure the concentration of tubulin in various samples, we will add an anti-tubulin primary antibody. Now anti-tubulin primary antibody, and it will be also be mentioned whether antibody has been raised, which animal the antibody has been raised, goat, mouse, or rabbit. So that will be the primary antibody. The secondary antibody is specific to the primary antibody. The secondary antibody will be anti-goat or anti-mouse or anti-rabbit. That is how secondary antibodies are named. And this secondary antibody will have an enzyme conjugated to it. Okay, so when this antigen is present, this entire structure interaction will happen. Then you'll add a substrate, and then if the enzyme is present, this substrate will be converted to its color product, and this will give out a visual readout. So the amount of color produced will be a clear indication of the amount of antigen present in the sample. So this is the basic principle of the assay. So there are different types of ELISAs: indirect ELISA, sandwich ELISA, direct ELISA, competitive ELISA, and this method has been used to detect various diseases, hepatitis B, rabies, and HIV through blood serum, just to name a few diseases. So this is the procedure of indirect ELISA method. There is a known antigen that is bound to the wells of the microtiter plate. Then there's a step called blocking. So the blocking step is done basically to stop non-specific interactions, and this step is done using BSA or casein. Then there's a washing step, so that any kind of unbound antigen or non-interacting protein can be washed off. Then we add the primary antibody. Then again there's a washing step. This washing step is to make sure that the primary antibody doesn't stay attached to any non-specific interactions. Then we add the enzyme linked secondary antibody. The enzyme linked secondary antibody will go and bind to the primary antibodies. Again, a washing step is present also to ensure that there is no non-specific interaction of the secondary antibody to the plate, to the wells. Then we add the substrate of the enzyme, and then which gets converted to the product and gives out a visual readout. So one enzyme that is popularly used is horse radish peroxidase, and it converts TMB, this is a substrate, to blue color. In case of alkaline phosphatase, PNPP is a substrate which it converts to yellow color. So these are very popular enzymes that are being used in this case, and the results can be read out using a spectrophotometer or a spectrophotometer or electrochemical device. Basically, the readout can be read, and the amount of color produced is proportional to the amount of primary antibody bound to the antigen proteins on the bottoms of the wells. This is schematic. This is an antibody. We all know antibodies are Y-shaped glycoproteins, and antigens have these particular places known as epitopes, which is the place where the antibody-antigen interaction happens. This is a primary antibody that is specific to specified to a specific antigen. This secondary antibody specific to a primary antibody. Very important. Secondary antibodies are always specific to the primary antibody. They are anti-goat, anti-rabbit, or anti-mouse, depending on which animal the primary antibody has been raised, and the enzyme is conjugated to the secondary antibody. Basically, to increase the signal, the secondary antibody step is used for signal amplification. So in indirect ELISA, there are two antibodies used. As I've already said, the primary antibody will detect the antigen of interest, and the secondary antibody will detect the primary antibody. This is the schematic. The antigen is attached to the bottom of the well. The primary antibody is attaching to the antigen. The secondary antibody is attaching to the primary antibody. The enzyme converts the substrate to the product, and a visual readout is seen. In case of sandwich ELISA, it is basically used to detect the presence and concentration of a specific antigen. So as the name suggests, in sandwich ELISA, an antigen is basically sandwiched in between two different antibodies, two layers of antibodies. Okay, so the antigen that is to be measured, this one, needs at least two different epitopes that will bind to the two different antibodies. So the antibody is attached to the well. This is called the captured antibody. Then we have the antigen of interest. This has an epitope for this captured antibody, and thus this interaction happens. This antigen is another epitope for the detection antibody, which we add after that, and then this detection antibody also attaches to the epitope of this antigen. Thus this antigen is now sandwiched in between these two antibodies. Then we add a secondary antibody that is linked to the enzyme on top of it, and then the substrate gets converted to the product, which gives a visual readout. That is why this is known as sandwich ELISA. In case of competitive ELISA, this is another version of ELISA where our antigen of interest, there's a competition between the unlabeled antigen or antigen of interest and a labeled antigen. Okay, so basically the color or the signal produced in this case will be inversely proportional to the antigens of interest. In the previous two cases, the color or signal produced is always directly proportional to the amount of the antigen of interest. But here what happens, we have an unlabeled antigen and we add a labeled antigen. Labeled antigen is the antigen already linked to the enzyme. Okay, so if the concentration of labeled antigen is more, then there's more chances that the labeled antigen will go and get attached to the antibodies that is captured on the surface of the microplate. If the concentration of unlabeled antigen is more, then chances are more that the unlabeled antigen will go and get attached to the different antibodies, captured antibodies on the microplate. Okay, so depending on that, after we add the product, the enzymes will convert the substrate to product. So there are obviously there will be washing steps in between to remove any kind of non-specific interactions. So if the concentration of labeled antigens is more substrate will be converted to more product, and the color will be, there will more visual readout. But if the concentration of our antigen of interest or unlabeled antigen is more, then less substrate will be converted to the product, and there will be lesser color produced. That is, which is why in this case in competitive ELISA it is inversely proportional to the antigens of interest in the sample. This is a schematic to show you the 4 different types of ELISA that we have talked about. Direct ELISA, where the primary antibody is already conjugated to the enzyme, signal readout basically. Indirect ELISA, because there are 2 antibodies. So indirect ELISA is preferred because there is signal amplification in this case. Okay, and there is sandwich ELISA. The name is self-explanatory, and then there is competitive ELISA that I've already talked about. So what happens? This is an enzyme, horseradish peroxidase, that is basically linked or conjugated to the secondary antibody, which transforms a substrate to the colored product, which gives the visual readout. So these are 2 of the references from 2 papers which first came up with the use of these 2 enzymes, alkaline phosphatase and horseradish peroxidase, and these papers are very important and are worth reading. So this shows a schematic of how the entire reaction occurs, not schematic exactly, the result section. So in any kind of experiment that we do, we always have a positive and negative control. For example, the positive control will sure shot give a color because it definitely contains the antigen of interest. The negative control is not supposed to give a color because it is devoid of the antigen of interest. And then we have our different samples. So all these controls are taken so that to minimize the error and to basically have more confidence in our results. For example, we can see the positive control is showing a very deep color and the negative control is showing a very light color. And then we have 3 different patient samples. Even visually we can see and say that patient C probably has that antigen in very high concentration that we are measuring over here, that analyte in very high concentration. Patient B also has that analyte but to a lesser concentration than patient C, and patient A is almost comparable to negative. So patient A is almost devoid of the analyte that we are trying to measure. Obviously we need a microplate reader to do a proper quantification of the data, but even visually we can see this. So after this we can go to a microplate reader and visually quantify our data as well. So these are the 2 important enzymes that are used in this ELISA technique, and there are some benefits of using enzyme probes. So enzymes have been utilized as detection probes for many decades. For many uses, a variety of enzymes have been used to detect foreign protein, antigen or other biological agents after being conjugated with another protein, usually an antibody that binds to the target, which is usually an antigen. So what are the benefits of using an enzyme probe? So there are several advantages. Long shelf life. The enzymes horseradish peroxidase and alkaline phosphatase are relatively stable when stored appropriately. Obviously good sensitivity. When the chemical reaction occurs, the resulting signal output can be relatively easily detected, which allows for identification of even very low levels of target protein or antigen. And in addition, there are also other methods available to amplify the signal so that the test is more sensitive. Finally, because of the high turnover of the enzymes, this also results in more enzyme binding to the protein, and the signal emission can be detected for a long time. Then it's a versatile system. Today there are many substrates commercially available that can be used with these enzymes, and obviously the probes are also commercially available as well. There are certain negative aspects as well, as for example we need another substrate for these enzymes to work. Only these enzymes in the system, that will not be enough for the ELISA technique to work out. We need substrates, which is an additional reagent that we need, and obviously there might be some background signals, that also non-specific background signal that might come, but obviously the advantages outweigh the limitations. Okay, so these 2, alkaline phosphatase and horseradish peroxidase, are very commonly used probes in this kind of study. And the horseradish peroxidase basically catalyzes the conversion of various substrates such as 3,3' diaminobenzidine or DAB, then 3,3' 5,5' tetramethylbenzidine TMB, which is very easily available. All these substrates to a colored output for our so that we can visually see it. For example, when HRP is conjugated to an antibody, it can be used to detect trace amounts of a specific protein in a Western blot as well. So the antibody provides the specificity to locate the antigen of interest, and the HRP enzyme in the presence of a substrate generates a detectable signal. And HRP is also used in immunohistochemistry and ELISA because it generates colored compounds. For detection of an antigen or protein molecule, HRP substrates have been designed so that they will generate a chemiluminescent, chromogenic, or even fluorescent signal upon oxidation. Okay, and it has quite small size compared to other enzyme conjugates, and this small size is also advantageous as it allows for more effective penetration into the sample cells and decreases the probability of interference with the conjugated protein function. And it's a high turnover. So the other key feature is that it's high turnover, and this results in an efficient reaction, producing an abundance of reactant products in a short span of time at normal pH. Okay, and even there are studies that show that HRP bound to IgG is much more efficient than alkaline phosphatase because of constant conjugate formation due to its higher specific enzyme activity. There is less steric hindrance, it is stable, and it's also cost effective. Okay, alkaline phosphatase is another enzyme that is used in case of ELISA. Okay, and that basically it also has some particular substrates like PNPP, which is paranitrophenyl phosphate. This is a particular substrate that will give rise to a colored product when the enzyme reaction occurs. Okay? So these are the two enzymes that are very popularly used in ELISA. So this is the answer. So the application of ELISA, there are many applications. There is serum antibody concentration, detecting potential food allergens, tracking the spread of disease, detection of antigen antibodies. All of these can be done very easily in the ELISA technique. So the answer of this question is A, PNR, that is alkaline phosphatase, and horseradish peroxidase. These two are very popularly used enzymes in ELISA. And for more information regarding this technique, you can go and watch this go and do this course, NPTEL course on analytical techniques in biotechnology by Dr. Ashwin K. Sharma, and there's a lecture that is already on YouTube, so you can, to learn more about this technique and all the details, you can go and watch this YouTube video as well. Thank you.